The neurobiology associated with Down syndrome. Down syndrome, also known as trisomy 21, is a genetic disorder that results in cognitive and neurological dysfunction. It is due to an extra copy of human chromosome 21. A chromosome is a DNA molecule that contains the genetic information of an organism. This extra chromosome causes gene expression in the brain to be altered. This gives rise to subnormal intellectual functioning, which includes deficits in learning, memory, and language. The World Health Organization estimates that the incidence of Down syndrome is between 1 in 1,000 and 1 in 1,100 live births worldwide. In Australia, the occurrence is 1 in 1,150 live births, which is 260 births each year. Fortunately, there has been an increase in the life expectancy of individuals living with Down syndrome. In Australia in the 1950s, the life expectancy was 15 years, a number which has now increased to 60 years. The age of the woman conceiving the child positively correlates with the chances of having a child with Down syndrome. For a woman conceiving a child at 20 years of age, the incidence is 1 in 1,400. Contrastingly, for a 45-year-old woman, the incidence increases to 1 in 32 live births. Neurobiology refers to the study of the nervous system and the brain. The remainder of this video will focus on the neurobiology of Down syndrome, that is, how the brain is affected in a person with Down syndrome. The exact reason for cognitive impairment in this condition is unknown. However, we can explore how the brain differs due to Down syndrome. There are multiple anatomical differences in brain development. Postnatal development. At birth, the brain weight of a baby with Down syndrome is at the lower end of normal range. However, by 2 to 6 months post-birth, 60% have a brain weight below normal, and by 2 years, 93% of babies with Down syndrome have a brain volume below the normal range. The following structural differences are observed. The hippocampus has an increased level of apoptosis, which is cell death. The hippocampus is a structure in the temporal lobe that has an important role in memory and in learning. Also, disruption to neurogenesis, the formation of neurons. Neurons are cells that allow for the transmission of nerve impulses and thus spread information throughout the brain. The third difference is altered myelination. Myelination enables nerve cells to transmit information faster and allows for more complex brain processes. Childhood brain development. Children with Down syndrome differ in the density and number of myelinated fibers in their brain. The myelination process is very important for healthy brain functioning, as it allows for information in the brain to be sent and received quickly. Delayed myelination was found in young children with Down syndrome. This was most apparent in the fibers connecting the frontal and temporal lobes. These delays are most prominent in the last area to be myelinated. This is the dentate gyrus. This structure plays an important role in memory and learning. These delays in myelination may result from weaker inputs to the hippocampus and/or alteration in the hippocampus itself. Brain development in adults. Adults with Down syndrome have a reduction in brain volume, including reduced overall grey and white matter. Some structures in the brain are also smaller. These include the cerebellum, which is located at the base of the brain and is responsible for coordinating voluntary movements such as speech, posture, balance, and coordination. The frontal lobe also appears smaller. This is located at the front of the brain and is responsible for cognitive skills such as emotional expression, problem solving, memory, language, and judgment. The frontal lobe can be thought of as the control panel of our personality and our ability to communicate. The temporal lobe is located at the bottom of the brain and contains the hippocampus, a structure responsible for memory and emotional responses. Reduced depth in sulci has also been observed. The sulci are the narrow grooves that can be found on the brain's surface. They function to increase the surface area of the brain, allowing for more neurons to fit into the cortex so it can process more information. Narrowing of the superior temporal gyrus has also been observed. This is located in the temporal lobe. It has a role in both verbal and non-verbal communication. Therefore, it participates in high-order auditory processing of speech. Also observed is reduced fusiform gyrus, which is part of the temporal and occipital lobe. This is located at the bottom of the temporal lobe and has an important role in object and facial recognition. Unexpectedly, the hippocampal gyrus appears larger in Down syndrome than in the typical population. This surrounds the hippocampus, a structure that was mentioned earlier. Electroencephalography, also known as EEG, is a device used to monitor electrical activity in the brain. When monitoring a patient with Down syndrome, decreased synchronized alpha rhythms can be observed. Alpha rhythms are present when a person is waking. This suggests impaired neural synchronization, meaning that the brain waves are not synchronized. As individuals with Down syndrome age, the differences in their brain become more prominent and begin to show signs of early onset Alzheimer's disease. In patients with Down syndrome, signs of Alzheimer's disease is present decades before it's usually found in the typical developing population. Unfortunately, by the age of 60, approximately 75% of people with Down syndrome have Alzheimer's disease. Overall, the neurological differences in Down syndrome result in disrupted cognitive functioning. Scientists must continue to research inhibition or improvement methods associated with trisomy 21's impact on the brain. Hopefully, this will allow for prevention of Down syndrome or treatments to improve the quality of life of those affected by this disorder. Thank you.